Cervical screening does help detect cancers at an earlier stage, but as countries ramp up screening in the population, what are cost-effective options? What are screening methods that are accurate? And how will we avoid overdiagnosis, unnecessary referrals, and unnecessary treatments of lesions that would have never progressed to cancer? Joining me today, Associate Professor David Hawkes, Director of the Molecular Biology Unit at the Australian Centre for the Prevention of Cervical Cancer. Welcome, David. How are you today?
I'm good, thanks. Thanks for having me on.
David, you want to shed more light on the whole idea of cervical cancer? How does the virus, we know the human papilloma virus is one of the main reasons, 99% of cervical cancers are caused by the human papilloma virus, but how does the virus transmit?
Sure. Look, HPV is a human papilloma virus. It's a virus for humans that causes papillomas, which can be everything from warts up to cervical cancer. There's 229 HPV types, but only 12 of them are shown to be absolutely causative of cancer. And the way we transmit it is by sexual activity. So any activity where you can be intimate with someone else involving the areas like your mouth or your genitals will allow you to actually transmit it. And it can also go through secondary things. So fingers or toys can also help transmit the virus.
So we only consider about 12, which could be cancer causing?
Yeah, so there's 12 that are absolutely cancer causing. There's another group of 8 which are possible or probable cancer typing. We don't test for all of those because a lot of them, it's only a very rare thing. We tend to test for either a group of 13 or 14 HPV types, and the International Society for Papillomavirus has said you shouldn't test beyond those 13 or 14 types.
You know, David, it takes a few years for it to become cancer. What happens in between?
Sure. So pretty much everyone gets a HPV infection at some point in their life if they've had sexual activity. The majority of those, over 90%, will actually clear within 2 years. So the way we think of it is if you get an ulcer on your mouth, it can clear, it can disappear pretty shortly. However, if you have an ulcer that's caused by the herpes virus, it can clear, then it will come back and it will clear. And HPV is pretty similar to that. Once that infection is established, so generally after a year or two, it's got into your cells and it will be there for a very long time. It starts to replicate. It starts to create lots of new viruses and create the components of viruses like proteins. And there's 2 proteins that the HPV virus makes that can actually cause the cells to change over a very long period of time, 10, 15, 20 years, even longer, so that those cells become odd. Originally, they become a little bit odd, they look a bit strange, and then eventually develop into cervical cancer.
And do we know what triggers that change in behavior of the virus?
So it's a bit of luck. The virus will get exposed to the cells on the inside of the cervix, sort of inside, outside of the cervix. Now, if those cells get shed and got rid of while the virus is in them, you get rid of the virus. Certain HPV types are much more likely to stick around. So HPV 16, which accounts for 50% of the cervical cancers in the world, it doesn't really matter which area of the world you're in, whether you're in Australia or Malaysia or Ghana or Belgium, it accounts for half of all the cancers. That is much more likely to stick around than some of the other 13 or 14 types.
You've been involved in the transition to a HPV-based cervical screening program, and that's part of the WHO strategy, the 90-70-90 strategy. What's that journey been like for you?
So, look, we started primary HPV-based screening in 1st December 2017, as it's quite a long time ago now, and we'd moved from a PAP-based program, which had been working for 20 years and was fantastic. We'd halved the amount of cervical cancer we have in Australia. Once we moved to HPV-based screening, it was a switch. It was a different kind of sample. We had to use bigger machines. A pap smear is a person looking down a microscope, and in our lab, they would look at 50 pap smears a day. So if we're doing 1,000 a day, we need quite a lot of people. Whereas, as everyone has seen with COVID, with some of these big machines, you can process quite a lot of tests. But what we've also seen is we've seen that we're detecting things that we wouldn't have detected with the traditional pap smear. So The two major grades of cervical cancer are what we call the squamous cell, so the SCCs, and they're really good for detecting with pap smears. But there's another group called the adenocarcinomas, and even up to 20 years, we were never really able to reduce the rate of those cancers, although they are detectable with HPV. So HPV allows us to actually detect more of these infections, which could become cancers later on. And so we are actually detecting a lot of lesions that we would have missed. We detect them with HPV. We can't see anything with the cytology. We send it to an expert, they do a colposcopy and they find a lesion. So it's a learning. I would love to say that we've worked everything out and we know what it's going to be like for the next 30 years, but we don't, because again, this is an evolving technology. Plus, when somebody is 50, if they're treated for their infection, if they don't have any other reason to get a new infection, they're probably never going to need another treatment.
More doesn't always mean it's better, David. How do we avoid overdiagnosis and unnecessary fear, especially if we're going to go toward mass testing and we want 70% of the population to actually get tested? How do we avoid overdiagnosing?
Look, I think that there's a couple of parts to that question. The first is that more testing is not better. Look, I talk about, I've got a dishwasher at home, and if I wash my dishes when they're dirty, they're clean, that's fantastic. If I wash them 3 times a day, they're not going to get any cleaner and they will have some side effects. I could sort of chip the plates, moving them in and out of the dishwasher. It's the same with any sort of screening. We used to have a pap screening every 2 years because it wasn't particularly sensitive, so we needed to do it more often to be able to detect these precancerous lesions. With HPV, in Australia, we use a 5-year interval. So if you're HPV negative, you come back in 5 years, you can have another test. The WHO has a 10-year recommendation. Other countries, it varies depending on age. This is something that is still evolving, but we do know that if you're testing for someone every year, you might detect an infection that's clearing. And so if you test it every 5 years, those infections, particularly when people are younger, they'll actually go away on their own. You do not need to affect them. And again, 90% of those infections will fall into that group. So we know that testing more often is not something that gives any benefits. Plus, HPV is not like COVID. You don't get the infection and you get sick 2 days, 3 days, 4 days later. You'll get the infection, it might be 10 years, 15 years, 20 years. So there's this long window in which we can actually detect the infection and treat it before you're at any, before your risk of actually having cancer is going to be there. And the second thing which you touched on is that there's 229 HPV types. We could possibly test for all 229, but it wouldn't actually give you any benefit. And testing for more might get you to do things more often. You'd have to see doctors more often. You have to see specialists. And one of the mistakes we made in the Australian program is for the HPV types that were not the HPV 16 and 18, which were the two that account for about 70% of cancers. We tested people, and if they were positive, they didn't have any cell changes, they came back in a year. If they were still positive, we sent them to a specialist. However, that actually meant the waits for a specialist ended up being over 1.5 years in some places, which meant that people that did need to see the specialist, they did have changes, they did have a reason to have them investigated, had to wait. And then we looked at all that information, and what we found was that we weren't actually detecting very much disease. So we've changed that to look a little bit further, and people come back for another HPV test. And we're finding that that's actually solved the problem. So when people have something that is worrying, they can actually go and see the specialist and have it investigated sooner rather than later.
Do we have enough specialists to cope with this when we actually start screening so many people?
Look, I think that this is a really variable situation. So in Australia, I live in Melbourne. We have a fantastic tertiary hospital that has a lot of colposcopists about 400 metres from my office. However, when I'm out I've just come back from a place called Uluru, which is a big rock in the middle of Australia. The nearest specialist is probably 1,000, 2,000 kilometres away. that's going to be a change. So I think that not just every country, but every different region is going to have different challenges. But I think that the learnings of some of the early adopter countries, so Australia, the Netherlands, places like Malaysia are certainly helping us understand these, will allow us to actually work out how we manage these situations.
Okay, so we do pap smears, right? And then we've got the HPV test. What's the difference in terms of how you actually get a sample? I understand you have a vaginal swab and you also have a swab from the cervix. Could you explain that difference to us?
Sure. So just to explain that, I might take a step back. One of the things we have with the cervical cancer screening program is we don't want to detect cervical cancer. What we want to detect is the lesions before they become cancer. If we can detect them, it can be a small biopsy, a small sort of little surgical procedure, and then it's over. We don't have a cancer. We don't have to have sort of more expensive treatment. And I looked it up yesterday, and apparently in a lot of Western countries, it costs about $70,000 to treat somebody in the first 2 years of cervical cancer. So it is a very big cost. Plus, I think most people, I'm approaching 50. I've had a few friends who have had cancer, and that's a really big trauma to yourself and to your family and to your children. So we want to avoid cancer, and that's what cervical screening is trying to do. So with the pap smear, there's a very specific area of the cervix called the transformation zone that we need to get those cells from, and then we can look at those cell changes. With HPV, we don't need to get to that specific area. We can, but we don't need to. And for better or for worse, COVID has made this explanation a lot easier. When I first had my COVID test in 2020, they had a swab that went so far out, I think it hit about there in the back of my head.
It almost feels like it hit your brain.
Exactly. And that was because they were trying to get to where the virus was actually infecting and replicating. And then they realized the virus has to get out, and so it comes down your nose. And now if you go and have a test, as long as the swab goes into your nose, It's absolutely fine. And it's exactly the same thing with HPV. We don't need to get to the transformation zone of the cervix because it actually sheds down the vagina. So as long as we can swirl around, we'll be able to detect the virus. And because we don't need to look at those specific cells, we don't need to go for the cervix, and that allows things like the self-collection to be a really viable, and certainly in our experience in Australia, it's a much more tolerated. I wouldn't say lowered, but I'd say it's sort of an easier test for people to do, and it's certainly less invasive of their personal space.
I think just not having to use a speculum is a huge advancement for the woman. So there are many types of self-sampling devices. You just want to explain to us what the difference is?
Sure, absolutely. So with HPV tests and with COVID tests and with swabs and all the things, there's a huge variety. And that's just the nature of anything. If you go out there, you can see a lot of different cars. Now, what we're worried about with the swabs, with the test, is you can have a great car, you can have a Lamborghini, a really fast car, but if you're going to drive on those really bumpy dirt roads, it's not going to be the right device for you. Still a great device, but it's not the right device for you. So when we talk about HPV screening, particularly self-collection, what we're looking at is the combination of the device, how we get it from the woman to the lab, and what the lab does to test it. So we talk about a system. So this is the traditional device, and that's a broom or a cervix brush, and that goes into the middle of the cervix, and that will get those areas. It spins around, they take it out, they swirl it in that little vial, which has got an alcohol liquid in it. It's about 20 milliliters. That goes into the lab, and they process it. It's very easy, very simple, but you need to be able to see where in the cervix. So you don't need something with that particular design if you're not going to try and get to the right area of the cervix. This is the device that is probably the most common. So there's slightly different versions, but it's a swab. It's got an end. Most of them are red, a couple of them are white. And this is the one that's shown, the manufacturers of a few different HPV assays have shown works. So this is called a flock swab. Now, all that means is it's a type of swab. So if you imagine a Christmas tree, so with all the branches coming off, it can collect lots of things, but if you turn it upside and shake it, everything comes off. So that's what the flocked is. It allows you to collect quite a lot of material, but it also allows you to swirl it in a liquid and get that material off so that we can actually test it. So this is probably very, very similar to the ones that you had a medical professional stick up your nose and twirl around. Probably not quite as big. It's a little bit gentler. They tend to be a bit thinner, but it's often a flock swab as well. And then, at least in Australia, they swirled it in a pink or red liquid. And that's the liquid. I think they normally use about 3 milliliters, then goes to the lab. Very similar, but the only difference is because HPV is a really robust virus, that's why it infects so many people, you can just take a sample. I think we say turn it 1 to 3 times for 10 seconds. It's not very much. And you can just put it in this, and then that can go into the lab. Now, that's probably the most common way. That's the way that we use it in Australia. That is the way one of the big companies, the BD, that's the way they do it. There's another company called Roche, and they take the sample, but then they'll actually swirl it in this liquid for 20 seconds, and they'll take it out and they'll send that into the lab. So it's the same device, it's different ways of doing these things, but both BD and Roche have shown with clinical trials that will actually give you a really accurate result. BD has also used this device, which is called the Evelyn brush. It's made by the same company that makes the devices that we've been using for cervical collection for years. This is called the Evelyn, so this was used in the Netherlands and is still used in Denmark for their screening program. So the woman will insert the end into the vagina. This will go in, they'll spin it around, pull it back down, put that cap in. And a lot of the time they'll actually get these through their postal service. So they'll be able to just put it in a little bag and send it back into the lab. And then what will happen is in the lab, obviously they'll use gloves, they won't do it with their hands. They can actually just remove the end, that will go into liquid, they'll mix it up and they'll be able to test that sample. There's a cheaper version, which is more suitable for places that, the Netherlands and Denmark. In my Australian terms, I can probably go from one end of my city, it is probably further away than some parts of their country. So this is called the bead brush. It's the same head, but it just means that you can take the sample and put it through.
And, David, how long can it last? I mean, what if it's a long journey? How long will that sample actually be relevant?
Absolutely. So with the Roche method, when it goes into there, it's up to 3 months. With these, particularly with the swab, with BD, so it's at 30 days at up to 30 degrees Celsius, and 6 of those days can be to 40 degrees Celsius. Now, the problem I have with these companies is they're all European or US, and I'm Australian, so we like to challenge things. So in Australia, the hottest recorded temperature, which was last year, was 50 degrees Celsius. So we've taken these and we've put them at 50 degrees Celsius for 28 days, and it still works. And then we've taken it down to minus 20 because that's the coldest recorded temperature. And that's the temperature that you keep your things in your frozen part of your fridge. And that is absolutely fine as well. So DNA is really robust. The example I give is I read an article about 6 months ago. They were trying to clone a woolly mammoth that they got from some DNA, which has been in the ice for 50,000 years. So, but again, we just need to have a device and a way of transporting it, so dry or wet, using a test and knowing that you'll get a valid result. So it's that combination that will actually allow us to feel secure that the result we're getting is the same quality of the result as we get with a clinician from the cervix.
So David, you've spoken about the swabs, right? One is a swab, one is a brush. And for the HPV test, the self-sampling ones, you actually take a sample, you put it back and you send it off to the lab. But there are some where you need to put it into a liquid and send it back to the lab. What's the difference?
Look, again, I'll go back to COVID. Nearly all the COVID tests we had to put into a liquid because it wasn't DNA. It was RNA, which is not as stable. And so we need to make sure that's going to be safe. DNA, again, woolly mammoths 50,000 years ago, it is very robust. Different companies have approached different methods. So they both have pros and cons. So if you use the method used by Roche, where you take the swab and you swirl it and then you send that in. This is the same device that we use all of our cervical screening tests in. So we've been using this since 2017 for every screening episode. Our labs are familiar with it. We have all the different places to keep it. We know how to process it. It's very, very easy. It's not free, but it is very, very easy to use. And once the sample goes in there, it's fixed. It's not going to change. It's not going to break down. That's an advantage. The disadvantage is that if you have to send all of these things out, because they're alcohol-based, if you have more than 1 litre, so that's 50 samples, it's classed as a dangerous good for any air transport and becomes very, very expensive very, very quickly. The other thing is this particular one is methanol. There's another made by BD, which is ethanol, but also has some formaldehyde in it. So you don't want to drink it. So you probably don't want things like this lying around or anywhere where there's a risk of it catching fire. So that's the pros and the cons. This is the same vial we've used for HPV-based screening for a long time. It's really straightforward. It's also built for cytology. So this was designed so that it will keep all your cells looking like cells, so that when you make a slide. With HPV, we don't care about the cells. It doesn't matter whether they break down. So when we have a swab, if we send that in dry and it takes 4 weeks to get in there, a lot of your cells will have broken down. You won't be able to look at it under a microscope. But since you're not collecting from the cervix, that doesn't really matter. And again, the virus, the viral DNA isn't really affected by it. So the good point about this is that this generally is cheaper than the vial by itself. It's also obviously very easy to ship. So we have a box of 100. It's pretty easy. I've carried 400 swabs in a backpack going from one place to another just to really transport them. They're quite cheap, they're easy. They also last a lot longer. So I think when these are made in the factory, they have about 5 years before they expire. With those vials, it's 2 years. And by the time they get to our lab, because it has to come by sea, it's about 1.5 years. So we do have to worry about some of those things expiring. The dry swab or the dry device, generally, it's just easier and cheaper. And as long as you know that it will perform as well. Our anecdotal evidence is that doctors really like these a lot more. They don't have to do different things. They don't have to remember, the more and more complex things get, the more difficult it is, because if you've got the swab, but then you don't have the vial, then you can't test it. If you don't have the swab, you can't test it, but you need to have something. And so this is why the dry sample is probably more preferred. It's easier, it's cheaper, it's just more accepted, and there's less things that go wrong. And our experience is the less things you have to do, the less things that can go wrong. And I'm incredibly lazy, and so I don't want to deal with things going wrong. So simpler is better. But again, it will really depend. If you've got a program that has cervical collection that also looks at cytology, you absolutely have to use the liquid media. If it's HPV only and it's vaginal, the benefits of the liquid media in transporting it are probably not that great. There are some other liquid medias that are not alcohol-based, but they are still shorter expiry dates. They are more expensive. And some of them have issues that if you ship them in a plane, it gets below 0 and they can have problems with, crystallization and kind of clogging themselves up a bit. The swab is a swab. It's pretty simple.
So it is clinically validated?
Yes.
Okay. How are the results presented, David? By HPV type or positive or negative? Or how will I, who has done this test, how will I interpret those results?
No worries. So different countries have different ways of doing it. I'm quite simple and I call it yes, no, or try again later. So yes means you have a HPV. Now, some countries like Australia and New Zealand, they go into a bit more detail and they say if you've got HPV 16 or 18, which are the two that are much, much higher risk of cervical cancer, you'll actually just go straight for a specialist intervention. And with the other ones, we'll look at the cells of the cervix. Other countries, so we know that Scotland, and I believe the Netherlands. Just look at whether you've got any of those HPV types, and then you go through that process. The no is they don't detect the HPV, and that's great. And then you won't have to come back until you're reminded by your national program. And generally that's between 5 and 10 years. The try again is if we don't have enough sample, or there's something that's affecting our ability to test things. So the example I give is not really the cervical sample, it's the COVID test. And we often get COVID if someone's got a really mucousy nose. Sometimes that will actually, we can't actually run the test because the mucus blocks everything up.
How do we avoid unnecessary referrals?
Look, I think understanding the risks of each program. So we know that if somebody is underscreened, so that they haven't screened for 5 years or 10 years, they're at a higher risk of having a HPV infection, they're at a higher risk of having abnormal cells, and they're at a higher risk of having cervical cancer. In Australia, one of the things that's actually changed in our program is that if somebody is underscreened, they'll be sent to referral before somebody who is a routine screener. Also, if somebody's over 50, if they have something that's abnormal, we'll send them to expert sort of referral before we would somebody who's 25. Because again, what your risk is depends on, your age and a number of factors. For instance, if somebody who's 20 and who's been smoking for 6 months has a persistent cough, we will probably not think they have lung cancer. If somebody who's 70 who's been smoking 3 packs of cigarettes a day for 60 years, we would treat those people differently. And it's the same sort of thing with cervical cancer and screening.
Okay. But it does affect the younger people, David. So this time you might look at it a little differently.
Yeah, I mean, cervical cancer for those under the age of 25 is very rare, and it seems to be a little bit more complex. So the WHO doesn't actually recommend screening until the age of 30. And part of the reason is most people become sexually active in that probably 15 to 30 years. And so they're more likely to be exposed to new infections. So we've done some studies, they're sort of subsets, so they're not the entire population. And as many as 46% of people between the age of 16 and 24 have a HPV infection at the point we test them. With our screening program starting at 25, it's 25% of people had a HPV infection. By the age of 50, it's only 6%. But those 6% are more likely to be those persistent infections. They're more likely to be infections that have been around long enough to start causing changes in the cells.
Is this given you've been vaccinated or you haven't?
So
Does that make a difference as to when you screen?
It doesn't make a difference to when you screen. Australia has one of the most comprehensive vaccine programs, and we also had a catch-up program. So people up to the age of 26 could be vaccinated in 2007, which means we're in late 2023. The oldest people are starting to turn 43. However, that wasn't 80% of people. Even those who were vaccinated, it was only against 16 and 18. So those with the extended vaccine, we call the Gardasil 9, they weren't actually drawing our screening program till about 2030. So if you've been vaccinated, your risk of having HPV 16 or 18 is really much, much lower. But there are still, again, another 11 or 12 HPV types that can cause cervical cancer that we do need to screen against.
So David, once those samples get back to the lab, how long does it take to process a sample?
So different assays have different approaches. So there's the fastest assay that's been clinically validated, can give you a result within an hour. There's a high volume that can do 1,000 a day that will give you the first result within 2 hours. A lot of them around 3.5 hours from when you put it onto the machine to when you get the result. Some go as high as 5 hours. Basically, once tests get into the lab, they'll do what they need to do to make sure that they've got the right sample from the right person and prepare it to put onto the machine. They put it onto the machine and then they'll get the result off. Generally, it's the same machines as your COVID PCR tests. Our laboratory, we specialise in cervical screening, but obviously it's a global pandemic, so we did over 400,000 COVID tests. And all that happened, the way I described it to my family and friends is that we took off the espresso pod on our coffee machine and we put on the latte pod. And that's really as simple as it is. It's exactly the same machine.
So a lot of our countries have the facility, existing facilities. You just need to change the pod, as you described. What about the quality of the test result? How do you control that?
Sure. Look, this is a massive difference between, say, a rapid test, which we all do our rapid test for our COVID, and a PCR that's done in a laboratory, particularly a laboratory that's, working to the sort of the quality standards we'd want them to. When you do a rapid test, I went and did one with my daughter last week, and it looked a little bit strange. I've been doing this sort of work for a long time, so most people wouldn't have picked it up. And then I looked and realized it was expired 6 months ago. Within a lab, we have to understand every single thing used in a test, from the tubes to the liquid to the swab to the test to whether the machine's been serviced. All of those things need to be accurate and need to be done. If the swab expired yesterday, we can't use it. And so there is a lot of quality control. The other thing is we have these frameworks, which is like a structure that we have to do. So we have to run, there's not much good with a test that tells you it's positive. We don't know if that's a real positive. So we have to run a positive sample, and it tells us it's positive. We go, that's good. We also have to run a negative sample and see, yep, the negative's negative. And we have to do those sorts of things. Most labs also do what we call external quality. And what happens is a few times a year, they'll get some samples in the mail from another lab, and they'll test them, and they'll say, well, we found HPV here, we didn't find HPV there. And you give the lab the results, and then they'll tell you whether or not you got them correct. And again, it's just making sure that, you can take anything and run a test, but if you don't know that, something's broken in the machine, it's giving you a lot of false negatives, you won't know that unless you actually challenge your tests. We have a saying in my lab, we try and break stuff. We try and find ways that will work out when it's not working. And we do that every day to make sure that whatever result comes off our machines and out of our lab is the same quality as the test yesterday, it's the same quality as the test tomorrow.